This is the second lecture that you should listen to instead of class on Monday the 4th. This lecture is going to focus on the pathways of amino acid biosynthesis. So first, before we talk about the synthesis of individual amino acids, we have to look at the two groups of amino acids, which are essential and non-essential. And as I've said many times, you remember that non-essential doesn't mean not important. The difference between essential and non-essential is that the essential amino acids cannot be synthesized. So, no synthesis in mammalian cells. All right, so can't synthesize them in mammalian cells. We have to get them from our diet. Now, there are two amino acids that are kind of an exception on this essential list, and those are arginine and methionine. If you remember when we discussed the urea cycle in class on Friday, we did talk about arginine, that we can produce arginine as part of the urea cycle, but we require almost all the arginine we produce in the urea cycle for the urea cycle, and therefore there's no excess arginine left over for protein synthesis. A similar process occurs with methionine. Methionine is used to generate a methyl group donor. We'll look at that a little bit later in these slides. And so the vast majority of the methionine that can be synthesized by mammalian cells is needed for that production of the methyl group donor, and therefore there's not excess methionine to be used in protein synthesis. We also have some non-essential amino acids that are made from essential amino acids, like we saw on the previous lecture video, the connection between phenylalanine and tyrosine, that tyrosine is synthesized as part of the phenylalanine degradation process. So even though tyrosine is not essential, a deficiency in phenylalanine would also result in a deficiency of tyrosine. All right, so now let's take a look at some of the pathways we can use to synthesize these amino acids, keeping in mind also that the degradation pathway we looked at in the previous video does not always mirror the synthesis pathway. So amino acids can be synthesized from intermediates in glycolysis, in the citric acid cycle, and pentose phosphate pathway. Now this slide shows the synthesis of all amino acids, but we're really only going to take a closer look at the synthesis of the non-essential amino acids. So there's some intermediates to keep in mind for the synthesis of those non-essential amino acids in mammalian cells. 3-phosphoglycerate is used as a precursor for serine. Pyruvate gives us alanine. And from our citric acid cycle intermediates, oxaloacetate, aspartate and asparagine, and alpha-ketoglutarate is also connected to synthesis of several amino acids. So let's look at some of these reactions in a little bit more detail. So first we'll start with the amino acids whose degradation pathways and synthesis pathways are pretty much the reverse of each other. So if you remember from the previous video, asparagine, we could hydrolyze and remove an ammonia to give us aspartate, and then through a transamination reaction we could get oxaloacetate. So the reverse is true for synthesis. For all of these keto acids, they can participate as substrates in a transamination reaction to give us pyruvate to alanine, oxaloacetate to aspartate, alpha-ketoglutarate to glutamate. Now with the majority of these reactions, the amino group donor in this case is going to be glutamate. In this case, it could be another molecule of glutamate, or it could be an aspartate. So glutamate is going to be our amino group donor for the vast majority of these transaminations when we're looking at amino acid synthesis. Once we have aspartate, we had a hydrolysis reaction to remove the nitrogen. Adding the nitrogen is going to take a little bit of input of free energy, so we have an ATP. In this case, glutamine is our donor of our nitrogen group. So here's something to remember. When we put our alpha amino group on, the donor is usually glutamate. When we're adding an amino group to another carbon, the donor is usually glutamine. So glutamate's the donor when we're adding it to the alpha position, glutamine's the donor when we're adding it at any other position. Synthetase, this is a ligase, we get pyrophosphate released, and you know if this is a go on to cleave it, then this reaction becomes irreversible. And we have asparagine. In a similar manner, alpha-ketoglutarate, transaminate to glutamate. Now, glutamine synthetase. Now, this is a slightly different reaction, where here we're using the free energy of ATP to add our amino group on to make glutamine. But remember that glutamine and glutamate have much different cellular roles than some of these other amino acids, so we have slightly different mechanisms for synthesis. So now let's look at the synthesis of serine. If you remember from the previous lecture, serine and glycine can be interconverted through the serine hydroxymethyltransferase reaction. It's technically possible to use this reaction to synthesize serine from glycine, but the reverse direction is more common in the cell, so synthesizing glycine from serine. So how then do we synthesize serine? Serine is the only non-essential amino acid synthesized directly from 3-phosphoglycerate. We have an oxidation to give 3-phosphohydroxypyruvate. Transamination, again remember that when we transaminate and we're putting the amino group on the alpha carbon, that the amino group donor is going to be glutamate. All right, now we have 3-phosphoserine. And now all we need to do is take that phosphate group off to give us serine. Once we have serine, then as I mentioned, we can use serine to synthesize glycine. Now this interconversion of serine and glycine is also really important because it's used to produce a cofactor called tetrahydrofolate. So the next thing we're going to do is look at tetrahydrofolate and its role not only in the glycine to serine reaction, but in a variety of other reactions as well. So let's look at the structure of tetrahydrofolate. This is an important coenzyme derived from folic acid that helps with the transfer of one carbon groups through a variety of metabolic reactions. Folic acid is necessary in the diet to produce tetrahydrofolate, and we'll look at that synthesis in a minute. These one carbon groups can be carried on N5 or N10, or can actually be bridged between N5 or N10, and can exist in several different oxidation states like methyl, methylene, or formyl, and we'll look a little bit more at these individual groups and their particular role in the cell in just a minute. So here you can see the pathway from folate to tetrahydrofolate, and the important thing to notice in this pathway is that it is catalyzed by an enzyme dihydrofolate reductase. This enzyme is necessary to regenerate tetrahydrofolate from the intermediates. So in some reactions, tetrahydrofolate is converted back to the dihydrofolate form and then must be regenerated, and that requires dihydrofolate reductase, although it's also involved in this initial synthesis. So dihydrofolate reductase is a very critical enzyme to a lot of different processes, not only amino acid synthesis as we see here, but we'll also see it when we come to synthesis of nucleotides. Therefore, blocking dihydrofolate reductase is a way to block cell growth. In order for the cells to continually grow, divide, and replicate, they have to be able to synthesize amino acids and synthesize nucleotides. If that growth becomes unregulated, as with cancer cells, and you block that growth by blocking dihydrofolate reductase, then you kill the cancer cells. So these are some of the common drugs that are used to target dihydrofolate reductase. Methotrexate is a common chemotherapeutic agent that is used to block dihydrofolate reductase and therefore block tetrahydrofolate production. This blocks amino acid synthesis and also blocks nucleotide synthesis. So here are some of the particular groups that can be carried on tetrahydrofolate. So this, for example, is showing methylene tetrahydrofolate, where our carbon group is bridged between 5 and 10. There are different oxidation states that can exist. Tetrahydrofolate, again. The most reduced would be methyl tetrahydrofolate, and that is used in a single reaction to transfer the methyl group to homocysteine to form a carbon-sulfur bond. This is part of methionine synthesis. This methylene tetrahydrofolate, 5,10 methylene tetrahydrofolate, is used to form new carbon-carbon bonds. And then the most oxidized form, which would be 10-formyl tetrahydrofolate, would be used to form new carbon-nitrogen bonds. So the type of tetrahydrofolate determines the reaction that it participates in and the cellular process that it becomes involved with. So now let's look at some of the reactions involving tetrahydrofolate. So if we start with tetrahydrofolate, we can synthesize 5,10 methylene tetrahydrofolate through the conversion of serine to glycine. Remember, this is the reaction that could potentially be used to synthesize serine, but in the cell most often occurs in this direction to help us synthesize 5,10 methylene tetrahydrofolate. Once we have the methylene tetrahydrofolate, this can be converted to the most reduced form of tetrahydrofolate, so we're reducing. So we're oxidizing the NAD, or NADP, most reduced form, and this is the one reaction that 5-methyl tetrahydrofolate can participate in, which is homocysteine to methionine. Now this methionine serves as a methyl group donor, and we'll look at that in just a minute. And again, this is why methionine is considered essential, because even though we can synthesize it right here, this is all used for methyl groups and there's no leftover methionine for protein synthesis. The 5,10 methylene tetrahydrofolate is also a cofactor in a very important reaction that converts a ribonucleotide, a deoxyribonucleotide, DUMP, into DTMP. This is the only synthesis pathway, de novo synthesis pathway, to give us T. And as you know, synthesis of DNA requires the 4 nucleotides A, C, G, and T. Methylene tetrahydrofolate is converted to dihydrofolate. If you remember from the previous slide, dihydrofolate can be converted back into tetrahydrofolate through the reaction of the dihydrofolate reductase enzyme. Now, if we block dihydrofolate reductase with one of those chemotherapeutic agents like methotrexate, then we block synthesis of TMP, which blocks DNA replication. So this is a way to kill cancerous cells. Now, methylene tetrahydrofolate can also be used to synthesize some of our other tetrahydrofolate 1-carbon carriers. So here we have one with double bond, C double bond to the nitrogen. We can make a formamino, we can make a formal. We have 5-formyl THF and 10-formyl THF. And again, remember, this is our most oxidized form. This can be used to make formal methionine tRNA. This is our initiator tRNA in bacterial cells, but is also initiator tRNA in mitochondria. But it's also used for purine synthesis. And again, we get tetrahydrofolate that then, if we went back to the previous slide, could be used to make methyl tetrahydrofolate. So we have this constant recycling of these folate groups, although we do need a constant supply of folic acid to keep up with the demand for all of the different folate forms, all the different tetrahydrofolate forms. So as we've seen several times now, there's a lot of interconversion between glycine and serine. This is important for producing our methylene tetrahydrofolate, which is required for the synthesis of some of our important molecules such as TMP from UMP for the synthesis of DNA. Glycine, though, is a very important biosynthetic precursor. Glycine is used to make purines, porphyrins. So remember porphyrin rings, those are the rings in the cytochromes and in hemoglobin. Glutathione, which is important for reactive oxygen species. Creatine phosphate, which is important to serve as our reservoir for ATP. Serine is also very important. It's used to produce phospholipids, used to produce cysteine, and serine can also be used to produce pyruvate, which then can be a gluconeogenic precursor. So we saw this when we looked at amino acid catabolism. Now let's look at another role for amino acids along the theme of amino acids as important biosynthetic precursors, and that is the role of methionine in producing S-adenosyl methionine, which is our methyl group carrier. Now there was one methyl group transfer in one of the previous slides in the production of methionine from cysteine. That is the only methyl group transfer that does not use S-adenosyl methionine as the source of the methyl group. So first, down here at the bottom, we can see synthesis of S-adenosyl methionine, starting with methionine and ATP. Again, we get release of phosphate, which is helping to push the reaction forward, and we have S-adenosyl methionine. Then, in its role as a methyl group donor, S-adenosyl methionine, so here's the methyl group that's going to be donated, can react with a substrate to donate the methyl group, and the product we have is S-adenosyl homocysteine. Now, as we saw on the slide looking at the synthesis of methionine, S-adenosyl homocysteine can then be used to produce homocysteine, which can then be remethylated to give methionine, and the cycle continues. So on the next slide, we'll take a closer look at this methyl cycle. So let's look at that. This is the methyl cycle. So again, we're starting here with methionine that was derived from cysteine. So we have our methyl tetrahydrofolate to tetrahydrofolate. This gives us methionine. Remember, this is the only methyl group donation that occurs without S-adenosyl methionine as the methyl group donor. Tetrahydrofolate can then be regenerated into methyl tetrahydrofolate in the conversion of serine to glycine, and then in a NAD or NADPH dependent reduction. Methionine converted to S-adenosyl methionine, converted to S-adenosyl homocysteine, which can then be converted to homocysteine to restart the cycle. So a couple of important reactions to point out. One is the synthesis of methyl tetrahydrofolate from methylene tetrahydrofolate. Remember that methyl tetrahydrofolate is only used in this one reaction to convert cysteine into methionine. Therefore, if for some reason methionine synthesis is blocked, methyl tetrahydrofolate can build up and disrupt the whole methyl cycle and the whole tetrahydrofolate cycle. The other thing is that the enzyme that catalyzes methylene tetrahydrofolate to methyl tetrahydrofolate is in essence irreversible because of the NADPH concentration in the cell. This is also a very important regulatory mechanism. So the enzyme that catalyzes this step is really catalyzing the committed step in all of methyl group biogenesis. So its regulation is critical to regulate all one-carbon metabolism. So therefore we have feedback inhibition from S-adenosyl methionine. If S-adenosyl methionine levels are high, this is the only pathway to S-adenosyl methionine. So if we block this step, we don't make any methyl tetrahydrofolate, and therefore we don't make any methionine. So this is a feedback mechanism. If there is sufficient acetylmethionine, then this homocysteine, instead of being recycled into methionine, it can have two different fates. It can either be converted back into cysteine, or if this pathway is blocked, then homocysteine is excreted. So as we see here, there is a genetic defect in which this pathway is blocked, and therefore excess amounts of homocysteine are excreted, which can lead to mental retardation, damage to blood vessels, and problems in the eye. There are also other genetic disorders involving the methyl cycle, including either deficiencies in the enzyme that catalyzes the conversion of methylene tetrahydrofolate to methyl tetrahydrofolate. And this deficiency of this enzyme can be minor or can cause major problems in the cell. So we've already talked about multiple roles for glutamate, serving as an amino group donor, serving as a way for amino groups to enter the urea cycle, but glutamate also serves for a precursor for synthesis of a variety of different molecules, including gamma-aminobutyric acid, or GABA. This is a neurotransmitter. Glutathione, which again is involved in reactive oxygen species. Glutamate also serves as the precursor, serves as the precursor for putrescine, which is a polyamine. Putrescine is a very bad smelling chemical compound, similar to cadaverine. Both putrescine and cadaverine are produced when amino acids are broken down in living or dead organisms. So this is part of the smell that a body has as it decays, but small amounts of putrescine are also produced in healthy cells and can be attributed to processes that don't smell very good, like bad breath. So next, let's look at a few other important molecules synthesized from amino acids. The first is nitric oxide. So we talked about nitric oxide a little bit when we were talking about the reactive oxygen species formed from side reactions of the electron transport chain. An electron can be donated to nitric oxide to produce a free radical, but nitric oxide itself has a very important role in the cell. It's a vasodilator, has a very short half-life. It acts as a signaling molecule. So it's produced and broken down relatively quickly. It's produced from a variation of arginine that gives us nitric oxide and citrulline. Remember, citrulline is a component of the urea cycle. So here we go from arginine to hydroxyarginine and then to citrulline. Glycine participates in this as well, which gives us ornithine, which is also part of the urea cycle. So you can see how nitric oxide synthesis is very tied into the urea cycle. Also, creatine phosphate, which is, as you remember, important for our ATP reservoir. When ATP levels are high, we form creatine phosphate. When ATP levels drop, we can form creatine and provide a little bit of ATP for the cell. So S-adenosylmethionine, our methyl group donor, is used to produce creatine, which then gives us creatine phosphate. We have a non-enzymatic method as well, hydrolyzing that phosphate. Of course, this is more beneficial to the cell, but we can't prevent these non-enzymatic reactions. Most of you are probably familiar with the connections between tryptophan and serotonin. So tryptophan is converted through a series of reactions into serotonin. So this is part of the hypothesis that if you eat too much turkey on Thanksgiving, that you get sleepy because you have all this tryptophan from the turkey. Tryptophan produces serotonin. Serotonin is used to produce melatonin, which is the neurotransmitter that is active at night. But in actuality, you would need to consume so much turkey that your stomach would probably explode before you'd be able to significantly impact serotonin or melatonin synthesis. So the reason that you're sleepy after you eat a big meal is because your blood sugar is spiking and then dropping. And finally, tyrosine is another amino acid that serves as a biosynthetic precursor, and it is used to produce L-DOPA, which we go on to produce dopamine, norepinephrine, and epinephrine. So our catecholamines that we discussed, these things that signal our fight or flight, they would cause muscle cells to release glucose from glycogen, to synthesize ATP. They would cause liver cells to release glucose into the bloodstream so that the muscle cells could take it up. So all of these are derivatives from tyrosine. All right, so hopefully now you have a little bit of a background about amino acid synthesis. And on Wednesday, we will discuss some questions related to both of these slides. These questions will be available on Blackboard for you to look at before class on Wednesday. So please watch these videos, look at the questions, and come ready to discuss.